My name is Gregor Neuert. I'm an assistant professor in molecular physiology and biophysics, as well as pharmacology and biomedical engineering. The focus of my research is to study how cells sense dynamic changes in the environment. So we want to understand how cells, for example, that are in the human body, can sense changes that dynamically occur in the environment, but we want to actually model this in a cell culture context, and we believe there's completely new biology that can be discovered by doing this. Because current research mostly focuses on fixed environments, which are not necessarily physiologically so relevant. Mainly we use microscopy methodologies to image live cells or fixed cells. We also use flow cytometry to measure many millions of cells at once. And also we use computational data analysis tools and modeling approaches to describe the data and model the data. So what attracted me is that there's a complete area of research that we do not really cover if we just look at cells in constant environments. And if we would look at cells in a dynamic environment which more closely resembles what's going on in the human body, we think we can discover completely new biology, and it's this new area frontier that attracted me. So basic science is important to really understand fundamental mechanisms about how biology and human diseases work. And without this basic understanding, it's very difficult to actually treat human diseases and maybe even develop cures down the road. And there's many open questions in biology that never have been addressed, and that need to be addressed first from a basic science perspective, before we can actually go and talk about treatments or drugs for human diseases. So we have two success stories. One is a more computational study, and this is if we generate a lot of data and we want to computationally model this, the question is how is this actually done? And we made some fundamental discoveries of what are limitations of current modeling approaches given new data sets that are generated. And this has implications for many different studies in biomedical research. And a second story that we are working on right now is to study how cells sense environment. And we found a new mechanism of rate sensing threshold, where the cells have a mechanism that enables them to sense how fast the environment changes compared to how much the environment changes. And this is a very fundamental process which we believe can be found in many different biological processes in cells. The advice I have for graduate students is to be excited about science, follow your passion, learn really the fundamentals in terms of biology, but also quantitative approaches, and definitely learn computer programming.